Good afternoon, everyone. So my presentation today will be about the work that FIND and its partners are doing in developing a new rapid diagnostic schistosomiasis diagnostic test to support national control programs to monitor the impact of their treatments, as well as for reassessment mapping. So somewhere on this line sits on the x-axis sits national control programs for schisto. And at each stage, there are specific diagnostic needs, and you will hear more from my fellow presenter, Lieven, on this later on. Now, the majority of the work that has been done in the red zone has been done using microscopy-based methods, CATs and urine filtration. Yet, as we move towards the right of the triangle, towards the green, that's towards elimination, there will be new diagnostic methods that are needed, that the ones that we've used until now in their current formats will no longer be useful. Most of you probably in the audience are aware of the advantages and disadvantages of the current diagnostic methods used for schistosomiasis, based on which we provide treatment, or ministries of health will give treatment. I'll go through these very quickly. The advantages are they're highly specific for the disease that we're looking at. They're field deployable, easy to use. They provide quantitative results, but also not only are you looking for schistosomiasis eggs in the stools, you're also looking for soil-transmitted Helminths. Unfortunately, though, the microscopy-based methods, they lack sensitivity. They're time-consuming. You have to read multiple slides, collect samples over multiple days, and also juvenile infections cannot be detected. So what this means is that currently microscopy underestimates true prevalence, which can lead to premature stoppage of MDA programs, especially when the intensity of infections are very low. And by stopping prematurely, it leads to recurrence of infection to original levels soon after. However, as Lieven will show in his presentation, there are current methods to try to improve this microscopy tools as well. So FIND and its partners are developing an easy-to-use, accurate and affordable schistosomiasis rapid diagnostic test, which is looking to detect circulating anodic antigen, the CAA. CAA is continuously secreted by living schistosomes, both juvenile and adult worms, and can be detected in the blood as well as the urine, and has comparable sensitivity to repeated microscopy. For this RDT, there will be no sample prep required, nor a reader for detection. And the intended use case is really to support the national control programs to monitor the impact of their treatment campaigns, but also to be used in reassessment mapping. To give it a bit of context, the FIND project on schistosomiasis started in 2018, and at the time it started with 4 partners, and over time this grew, both in terms of key schisto opinion leaders, research institutions, funders, and national control programs of endemic countries. We are now 8 partners involved in the consortium, and shown here on the map where we are, where we're working and with whom we're working. To give you a bit of a context of the project and the work that we've done since the start of the project, the first part is really about defining the target product profiles, the TPP. That's a document that is required by the test developer company, where it outlines all the attributes of the tests that is then used to produce the RDT or the diagnostic test that you want. So the TPP was developed, and then it was endorsed by the WHO Working Group on Guidelines for Monitoring and Evaluation and Reassessment Mapping back in at the end of 2018. And based on that, on the green line, the development of the CAA RDT started. And the first sort of prototype was available in November 2019, where it was field tested in Kenya on a small subset of individuals. At the same time, around that time, there was the new group, the Diagnostic Technical. Sorry, I got disconnected. Hopefully you can hear me back. At the same time, in November 2019, there was a new advisory group to the WHO NTD department, the DTAG. Now, the role of the DTAG was actually to set out TPPs for different diseases and identify the main use cases for those diseases. So the first meeting was in October 2019, and then they formed different subgroups for each diseases or group of diseases. So the Schisto DTAG subgroup was formed in April 2020, and we were tasked to do a diagnostic landscape of current existing diagnostic tools, as well as identify the priority use cases for schistosomiasis. And based on that, we developed a draft of TPPs, which are tool agnostic and biomarker agnostic. And they are currently under review with the main DTAG working group, as well as the WHO NTD department, before they will be publicly available for online consultation and for feedback from external review. So whilst this is going on, there is a development of the RDT. So after the alpha testing that was done in Kenya, the results from that study, as well as other studies that were continuing in the lab, the test developer continued to increase the performance of the prototype and created a version 1.2, for which the performance was verified in a lab-based setting. Now the prototypes have been shipped to the field study sites, which I will explain in a little bit, but in the meantime, we're also doing stability testing. And with R&D activities, it's also very important to look at the access component. And so we started doing some activities back in 2009 for the investment case, for which some of you who are probably attending the call today were interviewed, and to get some feedback on the Schisto RDT. So where are we now and where are we going? So currently the field evaluations have started in Kenya and in the Philippines. At the same time, there are some more fundamental research questions that need to be undertaken. And the results from both of these pieces of work will inform the further optimization required for the prototype. It will then be lab validated and optimized with a further smaller field evaluation before it goes into design lock, transfer to manufacturing, and validation verification studies in the laboratory setting to then move into the clinical validation trials. But all of this going on, we will start working in depth in the access activities in partnership with Merck, but also other partners. So where are we now? So this is the kit in which the prototype comes in. So you've got a chase buffer bottle. You've got the actual test device where you've got a sample well, and then the reading frame where you can read out the results. And then you've got a scorecard where you can score the intensity of your test band with this scorecard. How it works, you have to use 50 microliters of finger prick blood or venous blood. You apply it into the sample well. You wait for 5 minutes for it to be absorbed. After that, you will add 1 drop of chase buffer and wait for 15 minutes. And then you've got your results here. So the first 3 tests are positive with CAA present, are able to be detected, and then you've got a negative one here. So some results from the lab where the performance of the prototype was verified and actually met the TPP requirements. So here you've got the standard curve of spiked adult worm antigen, of which CAA is part of it, in naive blood. And what we were very excited to see, that at the 30 picogram per ml of CAA, we were able to detect a signal, which is the limit of detection for the test. What we also did was to compare the performance of the test with a very highly sensitive lab-based assay, which is the UCP-LF CAA assay, which is based out of Leiden University. Now, the antibodies that are used in the UCPLF-CAA assay are the same that are used in the prototype that we are developing at FIND with its partners. The UCPLF assay requires sample prep and a reader for detection, and it's fully quantitative. So what we were really proud of is the very good correlation between the testing of the samples with both assays. So that was very encouraging. So the field evaluations have started now in the country, so both in Kenya and the Philippines. So in Kenya, we will be going to 2 schisto-endemic sites, one that is for Mansenai and the other one for hematobium. And then we'll be testing in a non-endemic area for specificity. In the Philippines, one of the schisto-endemic sites will be for Japonicum, and we will also be testing for a non-endemic site for specificity again. So the study design, we will be collecting stool and urine samples for 3 consecutive days. And also on day 1, we will be testing venous blood and finger prick blood on the prototype RDT. Now, for stool and urine, we will be doing the Cato-CATS and urine filtration, as well as PCR on day 1 samples only. And for the bloods, so on day 1, we will be doing the testing on the prototype RDT, and then an aliquot of all the blood samples will be sent to Leiden University to be tested by the UCPLF assay, as well as aliquots from the urine as well. And a subset of RDTs will be reread 24 hours later, as it is one of the key attributes of the TPP for stability of the result. So finally, the question on access. And here it's really important to think about access as we start to develop as we start developing the test. What we don't want is that we have a test that is ready to be used and available, but actually there is no access plan. How's it going to be rolled out? Who's going to buy it? So we need to start thinking about this as soon as we start developing the tool. So whilst access is a term that encompasses many meanings, perhaps one of the key questions we need to ask ourselves is, how will the new tool be rolled out? What are the market barriers for such a tool, and how should we address them? So back in 2019, FIND did an investment case to evaluate and quantify the market potential for the launch of a schisto RDT in schisto-endemic countries. We did this by a range of expert interviews, collecting both quantitative and also we did qualitative data. And we wanted to look at quantify the market need and the market demand in 30 countries. And actually, some of you who are on the webinar today were probably solicited for this work. And what I'd like to share with you on this slide is a few highlights from this work. And what are some of the barriers we need to address as a community? So I'll give you four examples. The first one that was highlighted was around policy. So WHO guidelines for schisto are seen as the highest normative authority, but are not always strictly adhered to by countries, either by lack of clear guidelines or the lack of financial resources. So the question we should ask is, how will these guidelines around reassessment mapping and M&E evolve as we move forward? The next point is around funding. The majority of funding for schisto goes towards treatments, and only very little goes towards diagnostics. But from that, actually even a smaller proportion goes towards access and access activities. In addition, to ensure future sustainability of programs, we need to ensure domestic funding. And in this context for access, we're referring to the pathway to commercialization, which includes gathering evidence which you would obtain from conducting clinical trials, which is part of the data that is required as part of the regulatory dossier and registration dossiers that you need to submit. But not only are we looking at the pathway to commercialization, but we're also looking for post-commercialization, so post-marketing monitoring, which checks for the adoption of the tests. So the question here we should be asking is, how is funding landscape going to evolve in the future? The next point I'd like to raise that came up was around the registration process. National registration process and LMICs are complex and highly fragmented. All countries are different, each having different specific requirements. So how can we harmonize the registration process across countries? My final point will be, or my final example of topics that we need to think about in terms of access is around procurement and the distribution channels. There are limited players for procuring new tools, and the distribution channels are unclear. So what is needed to identify the commercial partner and then look at who and how the test will be procured and distributed? So thank you very much, and Cameron, I'll hand over back to you.